Reactome is an expert-authored, peer-reviewed database of pathways. Information is authored by expert biological researchers, maintained by Reactome editorial staff, and seamlessly linked to other bioinformatic resources. All of the source code, software, and pathway data is freely available for downloading and redistribution under open source terms. Reactome is an all-inclusive resource of human pathways for basic research, genome analysis, pathway modeling, systems biology, and education. There are several visualization and analysis tools that will allow you to explore the pathway knowledge in Reactome. The core unit of Reactome data model is the reaction. The concept of a reaction encompasses the transformation of entities such as the movement from one location to another and the interaction to form a complex, as well as the chemical transformations of classical biochemistry. Physical entities include nucleic acids, small molecules, proteins, and complexes. These entities participating in reactions form a network of biological interactions and are grouped into pathways. This generalization permits the capture of a range of biological processes that span signaling, metabolism, transcriptional regulation, apoptosis in a computationally navigatable format. The Reactome homepage provides quick, intuitive access to our data and tools as both features of the knowledge base continue to grow. The website has a comprehensive top menu bar that provides access to all of our tools and resources. It is accompanied by a sidebar to the left that provides access to basic, widely used tools for pathway browsing and data analysis, and panels to the right that give a thumbnail overview of Reactome, access to tutorials, recent news, and a view of a recently added pathway of topical interest. The pathway browser supports visual navigation and analysis of Reactome data. The browser consists of 4 key elements. First, the search bar at the top of the page queries the entire Reactome database. Second, the Pathways panel to the left provides a scrolling display of the Reactome canonical pathway hierarchy. Third, the Visualization panel to the right displays the interactive and dynamic pathway diagrams, permitting zooming, scrolling, and highlighting of events and molecules. Fourth, a Details panel below the pathway diagram with additional textual information about the events and molecules of the pathway. The molecular interaction overlay allows the display of proteins or small molecules interacting with the annotated protein components of a Reactome pathway. Interactors can be displayed for individual pathway proteins using the context-dependent menus in the pathway visualization workspace. It is also possible to overlay all of the interactors for all of the pathway proteins by means of the control panel feature. The pathway analysis tool analyzes user-supplied lists of genes, proteins, or small molecules and provides ID mapping, pathway assignment, and overrepresentation analysis. Pathway analysis results are presented in a tabular format with links to the pathway browser to visualize experimental data. Many types of identifiers can be submitted, reflecting the major gene and protein databases like UniProt and AndreGene, and the commercial platforms like Agilent, Affymetrix, and Illumina. The expression analysis tool is similar in design to the pathway analysis tool, but it will also accept numerical values, for example, expression, abundance, fold change, or a statistic, and shows how expression or abundance levels affects reactions and pathways in Reactome. Expression analysis results are presented in a tabular format and interactive pathway diagrams. The molecules in the pathway diagram are color-coded according to the data that has been uploaded. Like the pathway analysis tool, the expression analysis tool accepts the same gene and protein accession and numbers and identifiers. The species comparison tool allows the comparison of any of the species known to Reactome with human to find out which reactions and pathways are common to both species. Inferred events in Reactome are predicted based upon protein ortholog data from Ensemble Compara. After the species to compare events with human is selected, the results are presented as a table and as pathway diagrams. With our ability to present dynamic pathway diagrams and infer orthologous events in model organisms, we can now generate interactive model organism pathway diagrams. Furthermore, we've also developed the data analysis tools to function with these new inferred model organism pathways, providing the same level of support to the model organism communities as we've given to the mammalian biology researchers. Shortly there will be additional web tutorials to describe these other Reactome features and tools in more detail.